Today I'd like to welcome Stuart Strand and Neil Bruce presenting on their project ER-1504, Rhizosphere Bacterial Degradation of RDX, Understanding and Enhancement. Stuart, Neil, I'll turn it over to you at this point.
Okay, well, thanks, Deanne, Andrea, and Lisa. I'm here to present on this project, which was completed a number of years ago. The project team is at the University of Washington and at the University of York and at the University of North Carolina, as shown here. In the background, this project was initiated in 2005 in response to SOAN, as shown on this slide, and this was for the treatment and retainment of explosive and propellant-contaminated surface runoff from training ranges. And it invited innovative technologies and particularly mentioned phytoremediation in the SON, as well as microbial interactions with plants. And that was our focus in this project. So our technical objectives were to identify RDX-degrading bacteria in plant rhizospheres and discover the factors that control rhizosphere degrading population levels, to identify improved enzymes for the degradation of RDX, and to develop probes that could be used. So we accomplished some of these objectives and some we did not. The technical approach, as shown here schematically, was to set up exposures of grass and Arabidopsis rhizosphere to labeled RDX and labeled CO2, so that both carbon and nitrogen would be labeled. And then with isotopic probing of the rhizosphere, looking at the density separation. Let's see, I think I have a pointer. Oh, this puts a, can you see my cursor? There we go. Anyway, so density separation of the N15 DNA, and then screening or identification of the RDX degraders from the N15 gradients, and then looking at XPLA homologs. That was one of our intentions. And then also to vary the exudate composition using an Arabidopsis panel based upon the hypothesis that exudates could support as carbon sources the growth of bacteria using RDX as a nitrogen source. And we wanted to profile range grasses, the metabolic profiling of the exudates from the roots of range grasses. And then also we wanted to label the exudates from laboratory plants using C14, the exposure to CO2 of those plants, and then their exudates, their exudates would be labeled, and we could determine what the exudates were being taken up by bacteria in the rhizosphere. That was the intent of the project originally. So our results were to determine the microbial plant interactions of Rhodococcus 11Y and the incorporation of plant-derived carbon into ribosomal RNA, the microbial community of the rhizosphere. Also to determine, we also determined the lateral transfer of genes for RDX. We produced evidence for that. And we developed a high-sensitivity, high-resolution stable isotope probing method for quantitative TRFLP analysis of gradients. And then we applied that stable isotope probing technique to military training range soils that were degrading RDX. And we also had the results of co-cultures of an isolate from those soils, training range soils, that showed that nitrite could be taken up from the production of nitrite from RDX by XPLA-containing organisms, that nitrate could be taken up by other microbial members of the microbial community. Okay, So in the first project that I'll describe here, the Rhodococcus 11Y interactions with plants, which was done at York, and based upon research that suggested that phenylpropanoids comprise a significant fraction of the root exudates, that they could provide this is a little miswritten. They could provide a carbon source that would support the degradation of RDX as a nitrogen source in rhizosphere communities. A number of Arabidopsis plants, mutants, this is a mutant library of plants that have been found to have mutations in their plant root exudates. So these were accumulated at York. And then Arabidopsis plantlets were incubated using various methods, and their root exudates were extracted. And so the root exudates collected under different conditions did contain identifiable organic compounds, organic acids for the most part, in the root exudates. However, this slide is typical of replicates of root exudates extracted from Arabidopsis on all three plants grown under identical conditions as far as we could tell, and these were the root exudate HPLC traces from those cultures. And as you can see, they are distinctly different in major parts of the spectra, and this is actually one of the best replications that was achieved using triplicates and comparing them. So we could never get consistency in the root exudate profiles. Just to go back briefly, in addition, the root exudates, the mutant plants did not grow well in a liquid culture, and so it was difficult to obtain significant amounts of root exudates from mutant cultures. So that root exudate effort was abandoned at that point. So then the York group was looking at the inoculation of Rhodococcus rhodocus 11Y, a known highly active RDX degrader, and these are colony-forming units of the Rhodococcus after incubation in soil, and it did not do well as a rhizosphere colonizer. Also, it did not significantly add to the degradation of RDX in those cultures. So this is RDX remaining, and the addition of the bacteria had some effects, but not very much in Arabidopsis, and they were inconsistent in wheatgrass. So we concluded that 11Y is not a root colonizing bacteria. Okay. Excuse me while I'm struggling a bit with this. All right. So then the York group expressed XPLA, the gene, in Pseudomonas florescens, which is a root colonizing bacteria, a very well known bacterium. And they expressed the XPLA gene using shuttle vectors and expressed it in two strains of the Pseudomonas florescens. And these are all based on the PME 6010, which is a constitutive promoter. It uses a constitutive promoter so that the gene is on all the time. And they had to modify the codon structure, although that seemed to make a little difference in the activity. So here you can see that the Pseudomonas florensis was able to produce nitrite, as indicated by the formation of the red dye in this grease assay for nitrite. So, and this nitrite, of course, was released from RDX by the activity of XPLA in these Pseudomonas. So, here's a Pseudomonas containing an empty vector, and that is no XPLA gene, and there was no reaction, no production of nitrite. The Pseudomonas florescens, as modified with XPLA, was used was shown to degrade RDX, as you can see in this slide, by several of the different various vectors that were made. All had activity in the removal of RDX in pure culture. And again, a resting cell also showing RDX uptake. Again, RDX uptake by resting cells. And so then the fluorescence was introduced into the rhizosphere and of alfalfa and of Arabidopsis, and there was a degradation in this nodobiotic environment, that is only alfalfa and the modified Pseudomonas was present in this experiment, and there was a degradation and loss of RDX after exposure in this environment, as you can see again here. So this is the percent RDX recovered from the environment without the additional plants, and here with the XPLA present, much less RDX was recovered after incubation. And again here. This is actually showing very similar results. So this was done several times, and this is in SAM plots containing alfalfa with the Pseudomonas, and similar results for Arabidopsis. And here's a summary slide showing all of that work. Essentially, the RDX recovered, low being a good result, it was quite a bit lower with the Pseudomonas present. However, this seems to be mostly an additive effect. That is the plant uptake of RDX, which is a storage uptake. It does not degrade. And then with the Pseudomonas present and an empty vector, there's no significant uptake. But with the XPLA gene, there is activity of the fluorescens. And then combined together, you get the best result, but an additive result. And here's mass balance, and this was published in 2013. So in the summary of this section, the root exudate profiling had to be abandoned because there was a lack of consistency. And we also found that 11Y could not be used as a root colonizer. But fluorescence transformed was useful. I think I have an additional slide on this. Yeah, okay, so colonized by the transformed Pseudomonas, the rhizosphere was able to degrade RDX, and it could colonize the alfalfa roots efficiently too. After a few days of incubation, 35% of the colonies still contained XPLA, so the plasmid had some stability in the Pseudomonas. Of course, this was with RDX exposure. But under non-sterile conditions, the RDX removal was not enhanced in these rhizosphere incubations. So we still have problems with this transformed bacteria being competitive in the natural environment. Okay, moving on to the next section. This is Barbara McGregor's work at the University of North Carolina, and this is the incorporation of plant-derived carbon from C14 CO2 labeling into microbial RNA. And the object here was to identify the rhizosphere bacteria that are utilizing plant root exudate. is a fundamental science question that several groups are trying to advance. And this is a unique approach that use methods for separating ribosomal RNA so that they could be phylogenetically identified. The technique used magnetic bead capture of small subunit ribosomal RNA, and then single-stranded conformational polymorphism of non-denaturing gels of the rRNA, separate the rRNA, and then determine what's radiolabeled. That was the idea. Here is some of Barbara's work that shows probes that were developed as part of this work. These two probes, this BG probe and this alpha probe, and they were used to identify broad groupings of eubacteria on this phylogenetic tree. So these are very broad groups. They're not very specific at all. And here is some of control mixtures that were separated on these non-denaturing gels. And this is the rRNA of mixtures of pure cultures of bacteria. And helper probes were developed. These are non-labeled RNA that would help the labeled RNA from the sample bind to the to those probes for identification. And further work from Barbara, certifying her technique using pure culture mixtures and developing results. But the bottom line was that we were unable to obtain sufficient amounts of RNA for detection on her non-denaturing gels. And these are a couple of her gels that she ran using these soil samples, some from the training range, some from a munitions processing plant in Milan, Tennessee, and there was no bands identified using the phospho-imager even after 7 days of exposure. So no detectable labeled RNA. And this is further work, again confirming that. So and I'll just move on to the results. So they were not able there to identify bacteria that had taken up plant-derived carbon using these methods. Possibly by using heavier labeling and longer incubations and a larger sample size, enough would be accumulated. The group-specific probes met some objectives, but they really could not get into a significant specificity using those probes. So that objective was not met. Okay, I'm trying to advance here. Okay, so now the work at the University of Washington. When we first started this project, it was not known that RDX could be degraded on training range soil, and that the genes, the XPLA gene, which was known for RDX degradation, it was not known whether it was present on training range soils. So we added in this initial task to show that RDX could be degraded on training range soils, and that there was, and to look at the genes for RDX degradation. And in the course of doing that, we determined that there was significant evidence for lateral transfer of RDX degradation on extrachromosomal elements, specifically a plasmid contained in the Rhodococcus species. So this work is described here. So it's very common that recalcitrant organic degradation genes are on mobile elements such as plasmid. And there was some suggestion that might be the case for RDX degradation as well. So here we show that the near identical genes for XBLA and XBLB are across many species are on plasmids in two distinct species, the Rhodococcus rhodocus 11Y, and a rather more distant than other isolates at the time, Microbacterium MA1, which was isolated from the Milan base, the Milan processing unit by the UW team. So the fact that these genes are near identical, and then these more distantly related bacteria shows that they're more broadly distributed within the actinomycetales than previously recognized. So here are those species, the Microbacterium MA1 and the Rhodococcus 11Y up here. And so this distance is greater than the other species that had been found before the phylogenetic distance. The Microbacterium, as you can see, definitely grows on RDX as a sole source of nitrogen. And here is our money gel. This is shows the hybridization of the XPLA probe to the DNA from the Microbacterium and also Rhodococcus 11Y. That are these are the Microbacterium lanes. These are 11Y lanes. Similarly over here, and then this is a hybridization of the fragment to the XPLA gene. And these are on, I forgot to mention, these are on PGFE, pulse field gel electrophoresis. So they are separating out from the chromosome the plasmid DNA. So these are plasmid DNAs that are binding to these probes specific to XPLA in the two organisms. So this shows that the XPLA gene is located on an extra chromosomal element. And sequencing around the XPLA insert shows that the XPLA and B genes are surrounded by various transposase elements that would hypothetically make these elements mobile. Okay. And here it shows more generally the analysis of the fragment from that plasmid in the two organisms. There are some interesting processes around the XPLA and XPLB gene. And much of this is homologous, highly homologous between the two. So in other words, we found the XPLA gene, that it's been found in every organism that aerobically uses RDX as a nitrogen source, and has been recovered from RDX-contaminated soils and aquifers and other environments. And it's plasmid encoded, and it's part of a larger metabolic module surrounded by transposable elements. And the presence of those several IS elements in the vicinity also suggests that these could be readily integrated into a broader range of plasmids. So all of the elements for lateral gene transfer are here. And more recently, this work, this transposable aspect of XPLA has been confirmed and extended. Okay. So in order to do stable isotope probing with nitrogen as the stable isotope, it is necessary to develop a highly sensitive and higher resolution method in order to compensate for the lower separation of nitrogen-labeled bands compared to carbon-labeled bands. Now, C13 carbon has more generally been used for stable isotope probing, but we've shown that we can do quantitative stable isotope probing using this high sensitivity method. So we've developed a general protocol for the accurate determination of peak position by combining fluoroform for labeled primer with an intercalcating dye in order to link quantitative TRFLP restriction fragment analysis to the stable isotope probing. And the intercalcating dye can be used for real-time monitoring of amplification so that the amplification can be stopped during a linear range, part of the range, and before it is saturated. That is, the amplification reaction is saturated. And then the fluorophore label is subsequently employed to quantify the individual restriction fragments. This method differs from the TACMAN protocol, which has also been used for SIP of RDX N15 labeling. But that TACMAN requires an internal hybridization site. So this method simply relies on the two primers. The design of primers is greatly simplified with our method. And this is a schematic of the method. That we do a DNA extraction. We amplify it with this fluorophore, and we want to catch these amplification reactions at the midpoint of the reaction, rather than when they get up to this point, you see the lines separated. That's because there are different amounts of inhibition of the amplification process dependent upon the amplicon. So the qPCR product taken from these midpoints is purified and digested and then separated on a DNA analyzer. This is the T-Riflet step. And then the individual restriction fragments abundance are calculated from the qPCR of these products. And the copy numbers for each restriction fragment are plotted against the bland densities for each gradient of the SIP. Okay. So the results show that there was a very linear. This is the results from plasmid mixtures from several bacteria, and then when these mixtures, these are basically standards. Okay, so here's your copy number here, varying, and these are the regression of values of the copy numbers determined from with the plasmid mixtures. And as you can see, the R-squares are all very high for both area and height. So this is a verification of the method. So here we have 11Y being separated after growth on and also the Mycobacterium MA1 and E. coli are being given different sources of nitrogen, labeled and unlabeled, and you can see these are the results of this technique. You can see that by fraction collection, so this is the buoyant density in the cesium chloride gradient, and the fractions collected from that gradient, that they are rather beautifully separated from one another in these peaks. Now these are soils here, and let me recall, I forget what these soils were exposed to. Well, that's okay. We'll move on, and you'll see later gradients of the N15 RDX training ranges. Anyway, so the results from the from this technique development paper were that that this method worked very well, that it produces a very highly, going back, you can see that these are very highly resolved from one another. And there's the resolution and separation are very clear. And so using this method then, we advanced on to the stable isotope probing of military training range soils. And so several samples were extracted from a long-used bombing training range, and this was at Eglin Air Force Base in Florida. And these samples were screened for RDF degradation activity with and without exogenous carbon. So these samples were taken from around a target on Eglin, and this was vegetated soils, and we concentrated pretty much on vegetated soil samples, but we had a mixture of soil samples at different distances from the target center. Then we did stable isotope culture-independent stable isotope probing of the soil microcosms when incubated with fully labeled N15RDX. So this N15RDX is labeled both on the ring and on the nitro groups. So this is somewhat different from other published SIP N15RDX, since we had ring labeling as well. And then a quantitative PCR analysis was performed using the method I just showed you. Oops. Okay. And so here we see, this is an important slide. Here we see a number of samples, 23 altogether, and these were incubated with and without carbon added. We never saw any RDX degradation, and RDX added. We never saw any RDX degradation in these incubations without added carbon. That's an important point. The carbon, by the way, was a mixture of glucose, glycerol, and a third, I don't recall. But with carbon added, in some of these samples, the X indicating there was no degradation observed, the positive indicating that there was degradation. Here we also see, we also did the PCR amplification of XPLA, and in some we did not determine, yes, in some, here's a positive with RDX degradation and with measurable amounts of XPLA in several reactions. And you can see that only a few of the soil samples were able to degrade RDX. So there aren't very many X's here, or positives that is, and there are quite a few X's. So only about 30% were able to degrade RDX, and only in the presence of exogenous carbon. And in some cases we could see some XPLA, but we could not see RDX degradation. So you can see that there is, these are graphically shows you the results of the RDX incubations of these surface soils from the training range. And there was quite a lag of several days before RDX degradation began. This indicates that the RDX degradation, one possible explanation is that the RDX degrading population was very low initially in these cultures. And okay, so then when these soils were, the DNA was extracted from these soils after incubation with N15RDX, and at the midpoint and at the end of the RDX degradation in these soils, the DNA was extracted. It was run on cesium chloride gradients to separate the N15 labeled DNA from the N14 DNA. That is, the N15 labeled DNA being derived from organisms that presumably have taken up nitrogen from the labeled RDX that was supplied. And here are the results after analysis with our high sensitivity method. The restriction fragment 154, which was associated with the Rhodococcus species by sequencing, and it was the most abundant. It had a BD shift, the buoyant density shift, of 0.016, which was about the theoretical that is expected, of 0.016 to 17. from the in full incorporation of all of the nitrogen in the DNA as N15, shifted from the N14 peak. And here's the next most abundant species, an unidentified phylotype, and then the third most abundant, and these also have highly shifted DNA. A Variovorax species, another unidentified, a Variovorax is a gram-positive. This Mesorhizobium species, a gram-positive. The buoyant densities are beginning to go down now, so indicating less fully incorporation of N15 in the DNA. And then some other gram-positives, and here, and then much lower at this point, BD shifts. So these bacteria here are the most likely to have incorporated N15 into their DNA consistent, and this one here as well, consistent with degradation of RDX, but also possibly of the close uptake of a nitrogen-containing compound produced by the RDX degrading bacteria. Moving on. And here you can see the result in a heat map form. This is the unshifted spectrum across here. Oh, wait a minute. I don't know. You can see that, okay? Across here is the N14 gradient with no N15 present. And then this is replicates of the gradient for the fragment 154. This is restriction fragment 154, which is associated with the Rhodococcus. And you can see that that peak has shifted from the unlabeled peak. There's the next most abundant. That was an unidentified species with a restriction fragment of 273. And then the third, and so on down the line, and then the buoyant density shift begins to deteriorate compared to N14 as we get down into these restriction fragments. Okay, and these are ordered based upon the height and area of the restriction fragment on the electrophoretogram. Okay. Now, we also looked at the amplified XPLA fragment and from the buoyant density gradient, and here you see an N14 soil, and you can see that there. Now, these soils were taken from the ones that degraded RDX in our previous screening of these samples. They were mixed with some filler soil as well in order to produce enough soil volume, but that we knew that these soils were active against RDX. But at any rate, in the N14 label, you can see that this is the position of the XPLA functional gene. I'm sorry, I said that these were PHCR amplifications. These are not. These are amplifications out of the fractions, okay? So the fragments were not run on SIP, of course. These were amplification out of the specific fractions. So we could get some out of the N14, but after exposure to N15 RDX in these two replicates, the XPLA was shifted significantly. Okay, so XPLA genes from the DNA in those fractions were highly labeled. So the results were that the aerobic RDX degradation and the XPLA genes were observed only in a few of the surface soils. And this indicates that the RDX degrading potential was present at this location, at this target site, but it was heterogeneously distributed. So XPLA has made its way into these surface soils at this training range, but it is very heterogeneously distributed, just as RDX concentrations are themselves around target zones. And supplied with simple carbon sources, RDX degradation initiated after approximately 5 days and was completed within 8 days. Now, this delay suggested that these RDX degraders were initially present at a low concentration, or that it may be that competing nitrogen sources needed to be consumed prior to the initiation of RDX degradation. Now, we isolated 3 XPLA-bearing RDX degraders, the Williamsia and the Rhodococcus erythropolis, EG2A and EG2B were isolated from these egg-line soil microcosms. And we confirmed, remember, that the Rhodococcus species closely related to these isolated strains was involved, if not solely responsible, for the RDX degradation. So that Rhodococcus, which was closely related to these isolates in its phylotype or genotype, was the most abundant of the restriction fragments in RSIP. So we have confirmed that XPLA persists in military training ranges soils as well as in other environments. But we did reveal with the SIP analysis, reveal a number of organisms that could assimilate the N15 label after exposure, in addition to the Rhodococcus strains. And XPLA has never been identified in a gram-negative isolate. So this was intriguing. But we cannot rule out cross-feeding. That is a closely linked bacteria that could cross-feed on the nitrite released from RDX degradation, or perhaps from the NDAB. So this is the first. This study was the first to demonstrate RDX degradation in military training range soil. Now, we'd also looked at co-cultures of one of our isolates from the training range soils with nitrite degraders, and this is rather preliminary work, but it's suggested. So we isolated several species that were unable to grow on RDX, but most of which could grow on nitrite as a nitrogen source, a common activity in the bacteria. And then we co-cultured those isolates with EG2B with RDX as the nitrogen source. And here's what we got. So here you can see the red line with EG2B only, degrading RDX, growing, and producing nitrite. You can see the red line produced the most nitrite and grew similarly to others when they were mixed with it. So here it is mixed with the Rhizobium, mixed with the Burkholderia, and mixed with a Pseudomonas and Stenotrophomonas, which was not an RDX or a nitrite utilizer. Okay, so in one case, in most of these cases, we saw nitrite was greatly did not accumulate in the culture as it did with the Rhodococcus alone, and as it did with the Stenotrophomonas, which was not a nitrite utilizer. So with nitrite utilizers, they were able to take up the nitrite and grow, and in the case of the Pseudomonas, RDX actually was degraded faster in that co-culture than it was with the Rhodococcus alone. This has suggested that our SIP results have to be interpreted with caution, and many of the organisms that we found to be abundant there were possibly growing on RDX, on nitrite, released by the Rhodococcus or one of the other species that contained XPLA. Okay, so in conclusion over the whole project, the SIP findings support the abundance of Rhodococcus and RDX degradation in the new environment of training range soils. And RDX degradation may also occur as a result of gram-negative bacterial activity, but we cannot rule out the cross-feeding of nitrite to non-RDX degraders. And a large fraction of the training range soils around a target lack the genetic ability to degrade RDX. And training range soils incubated without carbon did not degrade RDX, even though they were from a rhizosphere. Our efforts to label rhizosphere communities with carbon labeled from plants failed. So we were not able to identify microbial communities that grow on root exudates and degrade RDX. Efforts to model plant root exudate profiles were confounded by high variability, so we couldn't continue that. RDX was not degraded by 11Y when 11Y was incubated with Arabidopsis plants. So this bacteria was not able to utilize carbon produced by the plant in the root zone. But a root colonizing Rhodococcus, Pseudomonas, genetically modified with XBLA, was able to fractionally increase RDX degradation when inoculated under notobiotic conditions, but not with non-sterile soil. I've taken the transition plan. We don't really have a transition plan to the BTP project. However, we do have some suggestions for further work. Now, training range soils have the potential for in situ remediation of RDX, and if two conditions are met, and that is the presence of RDX degrading bacteria and sufficient labile carbon to drive bacterial growth using RDX as a nitrogen source. So this implies that two techniques can be used that have been used in other areas of bioremediation: bioaugmentation, or the application of suspensions of live bacteria containing the genetic potential for RDX degradation, could be applied to these soils. And further, and a necessary condition, is that biostimulation by supplying carbon, labile carbon, could drive RDX degradation as a nitrogen source. Okay, so recommended research is to look at the viability of RDX degrading bacterial suspensions in the soil environment. Which species would be optimal? Which would survive best in the soil environment? Could encapsulation or other biopreservative methods enhance the longevity of RDX degrading populations grown in the lab and applied to soils? Can we grow up RDX degrading bacteria to large enough populations while retaining the RDX degradation phenotype? What is the best delivery method for the bacteria? Spraying, perhaps, of a wet solution, aerial spraying, or a dry powder on target areas, or perhaps some other more innovative methods? And how long will labile organic carbon applied to soil surfaces last? And finally, developing an empirical model of RDX degradation potential based upon the findings of the experiments, these empirical findings of how long these carbon supplies could last, how long the RDX degrading population could be present, and should be combined into an engineering model of this approach. Now here are our publications. We had 6 publications, some supported by this project and some directly the result of the project work. So that ends my presentation. Thank you.
Hi, thanks, Stuart. Are there any questions at this time?
This is Andy Anderson, and this is very interesting research, but I'm a little bit disappointed. I was hoping we could take Arabidopsis seeds, inoculate them, send them out, and then sprinkle them with sugar in a few days, and we'd have it working. It doesn't look like that's up.
I'm afraid that we were a bit naive in our approach to plant root exudates. It is a very complicated field, and I think it's the science and techniques of understanding exactly what sort of exudates plants produce and when, and under what conditions is not well understood. The lack of reproducibility meant that we could not interpret our results in any meaningful way.
Can I sort of interject a bit there, Stuart? So I think it's important to remember that we demonstrated proof of principle that it is possible to colonize root systems with bacteria that are engineered to degrade RDX. Now, the organism we used and the constructs weren't particularly stable, but I think with further research that could definitely be moved on.
Yes, and one thing I forgot to mention was that in those early days, those constructs did not contain XPLB. which greatly increases the activity of XPLA when in combination with that gene. So that would be something that could be followed up.
The fact that it could not be done in non-sterile soil really limits its use on an active range.
Yes, but this was the first effort to do this, and there have been studies in which bacteria have been introduced into label bacteria have been introduced into rhizospheres, and they've been more stable. So we could follow up on those and use those different bacteria as platforms for transgenic.
Yeah, I mean, part of the problem was that we were expressing a plasmid in that strain of Pseudomonas fluorescens, which really wasn't very stable. So after a number of generations, it was losing that plasmid. Whereas I think now with advancements in molecular techniques, it would be relatively straightforward to introduce those genes into the chromosome of a root colonizer. And I think the results then would be hopefully a lot better.
Yeah.
Thank you. That's the end of my questions.
I have a more, I guess, large-scale question. This is Hunter Anderson with the Air Force Civil Engineer Center. I'm kind of curious what you think, since you only really tested it out at Eglin, if like the composition of the range plants has anything to do with it, like in the northern climates where there's a greater proportion of things like sedges and cold season legume type plants, if there'd be more opportunity for the root colonizers to take hold and express the degradation activities you're looking for. I mean, is it site specific, or is it just sort of
well,
it works
it depends on which
everywhere?
We did in part of another project. We have looked at RDX degradation in Aberdeen, and soils from the Aberdeen training range in Maryland, and those soils had a similar heterogeneity of RDX degradation capability and XPLA presence. We did not look at rhizospheres. The Eglin soil was attractive to us because it's sandy, and that implies less difficulties in in DNA extraction. The Aberdeen soils are more loamy, and we may have had different results. I doubt it. I don't think that the root colonization the results were influenced by this, because they were not in Eglin soil. They were in laboratory environments. So there certainly are likely to be some regional effects on expression, but we haven't seen them yet.
All right, thanks.
Hey, this is Hans. If there's time, I'd like to ask, this is very productive, but it says to me it's really complicated. But the lateral transfer and the abundance of bugs that seem to do this suggest that maybe biostimulation is a pretty practical route to go. Can you speak to that?
As we know, the contamination is highly heterogeneous because it's particulate-based, and the occurrence of these bacteria is highly heterogeneous. So biostimulation applied over the area may be enough for degradation to occur around some particles that fell on soils near the training range that had perhaps that had XPLA or RDX degradation potential activities, the genetic potential for it in that particular soil area around the particle, or it may fall into an area that has none. An area like those samples that had no activity, even when biostimulated. So only about a third of our soil samples from around the target site had RDX degradation capability when stimulated. So if only stimulation was used, carbon sources, then you would expect RDX leaching to decrease by about a third, wouldn't you? Because only a third of the soil samples at any given target area at any point in time have RDX degradation potential, at least from our one sampling regime at this point. That's my reasoning on it. That may differ at other training ranges and at other targets for that matter. So that's why I suggested bioaugmentation as well, so that you eliminate that possibility and you increase your degradation. Well, hopefully you would increase your degradation from a third up more approaching 100% or 90%, something engineeringly more plausible.
Thanks.
Yeah.
Any other questions? Stuart, I just wanted to thank you again for taking the time to give us this briefing today. We do appreciate it. It is helpful for the program office as well as our review committee.